Hello, and thanks for coming to check out these review PowerPoints from which were originally developed for the Mammalian Physiology course, Bio 330. I've narrowed down the lecture quite a bit, and I'm going to talk about how what we learned in 330 directly applies to what you're learning now in pharmacology. So I hope you'll find these slides useful and will help bring a rounder and more complete understanding of the material that we're covering in the first few lectures, as well as down the line when you're looking at specific effects of different categories of drugs. Most drug targets in the body are going after proteins. Even drugs that have systemic effects and widespread systemic effects still are operating at the molecular level and are affecting how the protein functions, which implies how the protein is shaped. Like, for instance, if we were to somehow create this change in this protein, it would start pushing these helices around in such a way as to influence their relationship. And anytime you change the relationship of the structure in a protein, you're affecting potentially its function. Now, there's not a whole lot to say here other than that, yes, we have bonds and we can affect bonds in some cases. Some drugs do affect the ability of the bonds to form or how they relate and thus change shape. So the reason we went through those bonds in the first place was to tell you about pH and so forth, but now we can apply some of this to pharmacology as well. Again, just a refresher here about structure. We have a covalent structure and primary structure. We then have hydrogen bonding primarily and secondary structure, like to form this alpha helix or to form this pleated sheet here. These all can then fold into three-dimensional structure called tertiary structure. And I like this figure, and this is the main reason I kept this figure in here, was to show this heme group. And you could actually imagine this, instead of being a heme group, as some other pharmaceutical interacting with, binding with this three-dimensional protein structure. And that's quite often what's occurring with pharmaceuticals. You have a binding event, an association event, that thus influences the shape and thus the function. Quaternary structure, not a whole lot to say here. These are just different peptides that have associated with each other. Most proteins are, in fact, quaternary structures. Now, you can memorize thousands of drugs if you want to, and we'll talk about categories of drugs, but sometimes it's easier to approach and understand both effects and side effects from the functions of the proteins themselves. And so it may not make sense to you at this point, but as you get more and more familiar with the body and the body systems and more familiar with drugs, it's actually easier at times to think about it in terms of the protein structures. So that's why, and their functions associated with it. That's why I included this slide. So we may be inhibiting a catalytic function of an enzyme. We may be blocking transport of something across a membrane, blocking uptake. We may be altering or influencing signaling. For instance, caffeine is actually affecting signal molecules. It's actually serving as a mimetic for signal molecules inside cells. We can block or otherwise influence the effect of what are called receptors. Now, this is different from a drug receptor. Endogenous receptors in cells are designed to be perceivers, like, for instance, temperature or pH or gas or physical influences on the cells. We can talk about drug receptors, and drug receptors may or may not be specifically involved in cellular perception. It's just the protein that the drug binds. So receptors can be used, that word can be used in two different ways. Binding proteins, these are Proteins which can be chaperones, like for instance many of our most lipophilic drugs and steroids need a carrier protein for them to stay in the bloodstream properly. And so they do have a role in pharmacology. Immunoglobulins, we're not going to talk about them very much. Those are just basically immune response proteins, antibodies, etc. We may talk about them in terms of drug function at some point. Regulatory proteins, yes, sometimes we will influence genetic expression of certain things, or a response may influence genetic expression. So regulatory proteins are the coordinators and the controllers of cellular function. In terms of pharmacology, I'm not sure we're going to talk a whole bunch about structural proteins and fibers, but maybe. Oftentimes what we see is that if you're affecting a regulatory protein, that may cause a secondary effect on structural proteins down the line. For instance, let's say we give somebody an anabolic steroid. An anabolic steroid as a pharmaceutical would affect regulatory proteins, which lead to the expression of things like muscle fibers and actin, myosin, and so forth, thus affecting structural proteins. So it's good to know about those things. Just having that understanding will help make understanding drug functions a lot easier. In terms of this slide, protein interactions, we may talk about proteins binding with other proteins. We may talk about proteins binding with what's known as their substrate, their endogenous ligand. We could also talk about protein interactions with pharmaceuticals. It almost always involves a binding event that's non-covalent, but I'll give you a great example of one that is covalent, and that's aspirin. So it's not 100%, but the vast majority of drugs will interact with proteins at some binding site in a non-covalent way. And it is an equilibrium situation, and I'll explain that more a little bit later. Ligands, substrates, those tend to be the endogenous molecules that proteins bind with as opposed to pharmaceuticals. And we're going to talk about specificity and selectivity when it comes to pharmaceuticals. So yes, proteins do have specificity about what they will bond to and interact with or bind to. It's not exclusive, and that's kind of what selectivity is. There may be a given family of things that a protein will bind and interact with. And that's true both of ligands and substrates as well as pharmaceuticals. This is just showing it graphically what I was talking about just a second ago. That let's say here we've got an endogenous ligand. Let's say we have a pharmaceutical that is a mimetic of this ligand, a competitor, if you will, may bind in this binding site. And it is an equilibrium process. Depending on how high of affinity you have will affect the ratio of whether it's bound or unbound. Now in your book they're going to talk about the Henderson-Hasselbalch equation. I'm not going to have you calculate that. I just want you to understand the concept. The concept is that whether it's unbound or bound is a ratio, and that ratio is somewhat empirical depending on the affinity of the molecule for the protein. If it has a very high affinity, the ratio will be skewed over to the bound side. So you'll have much more likelihood of binding than not binding. If it has a low affinity, that will shift the ratio over to this side, where you're less likely to be bound. It's always a ratio. That's what the Henderson-Hasselbalch equation is all about. It's just describing mathematically that ratio of binding versus non-binding. Certainly lots of things can affect protein binding, whether we're talking about endogenous molecules or drugs. And so I'm including this slide as a way of bringing together the idea that we're going to talk about drug absorption and drug metabolism. These factors will play in pharmacology as well as in regular physiology. Sometimes the protein shapes are different. For instance, we know that Alcoholic families, families that tend to have high levels of alcoholism, may have forms of protein that don't metabolize ethanol the same way. And that's true with lots of different types of drugs, and may lead to certain types of sensitivities that we see, having to do with the different isoforms of the proteins involved with the pharmaceutical or molecule in question. The abundance of the proteins. If you don't have much protein, it can't be metabolized. We may talk about what we call induction, where, for instance, if you have someone who is a smoker versus a non-smoker, the smokers will often have much higher levels of proteins designed to eliminate toxins. We call that metabolic induction. And so that involves abundance. If proteins are induced, another famous example is grapefruit. If you eat a lot of grapefruit, you're going to have a lot more enzymes, which are known to be detoxifying enzymes, and so you have a greater abundance of those proteins. Whether those proteins are active and inactive, this is more to the point of inhibitors and activation. So I left this in here because drugs are fundamentally quite often involved in this activation and inactivation of proteins. They're modulators. So if you have something that is affecting activation and inactivation, that could be said to be a modulator. A chemical modulator is one that is affecting the function of a given protein. And we're going to talk about competitive and non-competitive, also known as allosteric, etc. So we'll get to that in the lecture, but it's something that you should have seen before. Physical factors. Oh yes, pH. For instance, we can modulate drug availability based on pH. We can modulate, or we can design drugs to be absorbed in certain areas based on lipophilicity, or the thickness of a membrane, or how much blood flow, exercise, etc. Temperature. Isoforms, again. So as we talk about activation and inactivation, it's important to understand that most of the time we're not talking about this. What we're actually talking about is more something like this, where we can dial up certain things in a graded fashion as opposed to an all or none fashion. So yes, we're going to talk about activation and inactivation, but it's going to be more of a modulation rather than an on-off situation. So extending from that, through activation and inactivation, we're actually talking about nuanced activation and inactivation. We call modulation. That means you can dial up things and dial down things in graded fashion rather than in all or none situations. And as a bad joke, this thing up here, if you know about music recording, is a fader. So with the recent Star Wars, I decided to put a bad joke in there, the Darth Fader. Now endogenously, we can have activators and inactivators for protein function. We call some of these things cofactors. And drugs may mimic or approximate cofactors or block the ability of cofactors to function. There are a number of situations where you may block absorption in the gastrointestinal tract by blocking the effect of a cofactor on a transport protein. So sometimes when we talk about cofactors, we may be talking about vitamins and nutrients. So when we get to those later chapters in the course, some of those we're going to be talking about in terms of being cofactors for proteins. Now we're starting to get into some more pure pharmacology. For instance, we're going to talk about competitive inhibitors. Competitive inhibitors are competing for endogenous ligands on binding sites, what we call active sites in proteins. So if this competitive inhibitor is, depending on its affinity, may be able to outcompete or partially compete with endogenous ligands. And that's again getting to modulation. Depending if this inhibitor has a high affinity, is very potent, it may be able to prevent the ligand from binding altogether. If it's a weak binding, if it has a lower affinity, it may be a more 50-50, or maybe a more 40-60, or 30-70, etc., etc. Depends on the affinity. Reversible and irreversible just is another way of saying that. If it's irreversible, it just means that it has a very, very high affinity, so that once it's bound, it's pretty much stuck there. We're not going to talk a whole lot about that in this class, I don't think. Again, when we talk about aspirin, we'll talk about an irreversible situation, but that's pretty unique. We will also talk about allosteric, sometimes referred to as non-competitive interaction. Allosteric is just saying that, hey, we have got an active site somewhere, and that allosteric modulator is working somewhere other than the active site. Whether it's an activator in this case, or an inhibitor in other cases. And here's an example of an inhibitor. In this case, it is not affecting the binding site. It's binding somewhere else other than the binding site. So lots and lots of drugs are allosteric inhibitors. And we're going to use that term quite a bit, whether it's a competitor or a non-competitive inhibitor. Most drugs are, in fact, inhibitors. So these are terms we're going to use quite often. This is just another figure showing that competitive versus non-competitive. So non-competitive is aka allosteric. Competitive binds or competes with the binding site. And depending on the affinity of one or the other, depends on the competition going on here. And it's always an equilibrium, it's always a probability or ratio, if you will. So this is just a list of terms. So we're summarizing what we've talked about to this point. We're going to be talking about all of these things at some point in the class. So it's nice to know these terms. I'd strongly recommend that you get conversant with these things during the course of this class. The more conversant you are with these terms, the easier it will be for you to present your material as well as understand drug action. And again, I've just included this figure here to show you, to jog your memory, to help you envision the pharmacological process at the protein and at the cellular level. As far as this slide is concerned, is just again getting at the idea that sometimes we measure effects in cells. So if we want to understand the efficacy of a drug, we oftentimes have to measure the effect upon the protein function. And so that's reaction rate. Reaction rates are dependent on how much ligand or how much substrate you have. And we can saturate. We can change the saturation point with different types of drugs. And that's where I'm going with this. So let's talk about the next slides. I think I have a figure coming up here. And let's show you what I'm talking about. If you have a given protein, it will catalyze a reaction and create some response. This is how we measure efficacy of drugs. Now, this may not have direct bearing on you in the clinic, but if you want to understand potency, you want to understand how we determine the effect of drugs, it's important to sort of understand what's happening at the protein. If you saturate the protein, if you add enough ligand so that the protein is working as fast as it can, that's called saturation, and that creates a condition known as Vmax. That is the velocity of the reaction, if you will, the maximal velocity for a given amount of protein. If we affect the protein in any way, we may affect how much concentration is required to reach that Vmax, or we may affect the Vmax altogether. Let's talk about that. This is really the take-home message. If you have a competitive inhibitor, you can eventually achieve Vmax if you keep adding more and more substrate. Okay, so if you add a given amount of competitive inhibitor, it's going to compete for the binding site. You can swamp that binding site by just adding more substrate, in theory. Okay, sometimes there are practical reasons that you can't, but theoretically you can. However, if we have a non-competitive inhibitor, it won't matter how much substrate you have, because you're not competing for the substrate's binding site. So you may have the same affinity, but you have now, with that non-competitive inhibitor, impacted the maximal velocity of the reaction for a given amount of protein. This is really, I bring this from pharmacology into physiology, and now you can see why I do that as we talk about these different kinds of drugs and the pharmacodynamics, if you will. Pharmacology is applied biochemistry. So having a sense of biochemistry improves your understanding of pharmacology. Having a sense of pharmacology understands why we would talk about something in biochemistry. So to sum up this part, we are talking about affecting shape, which affects function. So if we change shape, that affects perhaps the ability of a protein to bind its ligand or bind a substrate and carry out its process. We're going to use these terms quite a bit when we're talking about different aspects of drug metabolism, drug function, drug absorption, and so forth. So these are really, really important concepts in terms of understanding influence of drugs and so forth in this section. So I hope you found this lecture useful in that capacity. The idea that we're modulating proteins and biomolecules for functional repercussions.